All right. So, hello everyone. A very warm greeting to one and all. My name is Sri Gauri Krishnakumar, and I would like to welcome you all for the second webinar on microbiome in health and wellness hackathon. So, for those of you who are new to this webinar, welcome. And we are organizing a hackathon on microbiome in health and wellness this November to provide students with a platform to showcase their innovative mindset of problem solving. And in this hackathon, each student will have the opportunity to develop a mini project around the hackathon topic, form a team with at least two other students, and compete against others for mentorship, support from established companies, and long term interest from the business community. And the applications of hackathon are in the fields of obesity and precision nutrition, innovation and beauty products, and pregnancy. In our previous webinar, we discussed week one topics on microbiome business that will be covered during the training duration. We were joined by Mr. Ilya Brodsky, CEO of Pine Biotech, who discussed what the hackathon is about, what kind of opportunities are present for students and researchers who are participating in the hackathon. The participants of the webinar were also joined by Dr. Raghavendra Lakshminarayan, computational biologist and research consultant at Pine Biotech. He discussed what is microbiome, how bioinformatic approaches help understand association of microbial population with disease, and popular research publications that will provide ideas for students participating in the hackathon. In today's webinar, we will be first introducing the participants to the week 2 workshop on biotechnology, where we will be covering the topics on gut microbiome, health and diet, skin microbiome, beauty, comfort and aging, and vaginal microbiome, pregnancy, and femtech. So before we begin with today's webinar, for those of you who are joining for the first time, let me also take a brief moment to introduce you to the team behind the program. We are a US-based bioinformatics company who is working with multiple academic and commercial collaborators to develop easy-to-use analytical tools, and our mission is to make bioinformatics more accessible. And as a part of the hackathon, students will have access to the omics logic training that has been completed by over 24,000 participants from 187 countries in over 300 workshops. And due to this fast growth, our team is working with local and regional coordinators that are helping refine local program logistics and adapting them to the needs of students and researchers around the world. And this training has been completed by participants in 6 different specialization tracks that includes oncology, infectious diseases, precision medicine, neuroscience, data science for biomedical data, and comprehensive training on omics data analysis. As a part of the hackathon, students will also have access to the Omics Logic resources. But how does it help advance bioinformatics teaching? Omics Logic is a portal for practical and theoretical learning of bioinformatics. It takes a combination of training modules, data analysis tools, curated project datasets, and interactive sessions with mentors to give the student a clear path up the Bloom's taxonomy pyramid. No matter what the user's initial experience with bioinformatics is, Omics Logic is a useful tool for gaining a baseline knowledge of the theory behind bioinformatics, analysis of datasets, and introduction to basic coding languages like R and Python, or even beginning a data analysis project. This also comes with an access to Omics Logic courses, which includes courses on bioinformatics, precision medicine, multi-omics, including genomics, transcriptomics, metagenomics, epigenomics, data science that includes bioinformatics using R and Python, as well as cheminformatics. Users will be given directions to access the tBioInfo platform for bioinformatic processing and analysis of data, and the platform includes demo pipelines as well as data management and analysis cloud infrastructure to run the bioinformatic pipeline. And different stages of the analysis are performed in different sections on this multi-omics platform. So first of all, we need to sign up on the Omics Logic Learn portal. But before that, let me walk you through a brief demo on what a completed profile on the portal looks like. Once you sign up on the portal, these are the features that you need to update on your account. That is, a completed profile will have a profile image, full name with appropriate capitalization, link to your social media handles. This could be your Facebook, LinkedIn, Instagram, or ID, or even Twitter. A brief bio about yourself that will consist of your education background and research interests. The mentors can view your progress under the Activity tab. The courses you are currently completing will appear under the Courses tab. And finally, after the completion of your course, the certificates will appear under the Certificates tab. Once you sign up on the portal, you will also be able to view the various courses and example projects that will help you prepare for the hackathon. One course that I would like to bring to your notice, whether you are a beginner to the field of metagenomics or you are an intermediate or expert willing to refresh your metagenomic concepts, I would like to direct you to course 2, Bytes and Molecules. This course is intended for all those who are interested in understanding how the current revolution in biomedical data impacts biomedical research and human health. In this course, the lesson that will be of relevance for the participants will be Lesson 8, Introduction to Metagenomics. In this lesson, you will explore how different types of analysis can be used to understand the microbial world and practice several bioinformatic approaches to the analysis of the microbiome. Finally, to check your understanding, the lesson ends with a simple quiz. If you wish to proceed ahead and gain an in-depth understanding of the metagenomic concepts, you can opt for Course 4, Metagenomics. In this course, you will learn about processing 16S rRNA data with Data2, visualizing O2U abundance tables for diversity, using phyloseq-R package that can help build phylogeny-aware species richness, alpha diversity plots and beta diversity plots, and so much more. To help build these concepts, course use practical quizzes, hands-on demo pipelines, and R programming for data visualization. Once you gain a thorough understanding of the key concept involved in the metagenomics data analysis, one can start by going through one of the relevant example projects. The example projects are sourced from high-impact research publications that will provide students with the logic behind conducting a research topic. For instance, the example project on role of high-fat diet on obesity aims to understand the microbial diversity after feeding high-fat and normal diet in mice and decipher its role in obesity and underlying diseases. All the example projects follows a similar format where students are initially given an overview of the important concepts to carry out the example project. This is followed by identifying a gap in the literature and understanding the significance of the research question proposed. Once done, the students' knowledge are tested through quizzes and assignments. To provide hands-on practical experience, students are given step-by-step guidelines to building bioinformatic pipelines on the T-bioinfo server. And the section that will be of relevance for today's discussion is the metagenomics section. For this example project, there already exists a demo pipeline under the name Chow versus HFD. In this example, we are making use of 16S metagenomic pipeline. This is an example of how we will carry out the bioinformatics pipeline. The input files will consist of fecal samples collected from the mice, and our aim here is to analyze the microbial diversity. The pipeline is based on running a number of programs, including Data2, AVE, and phyloseq, and the functions of each of these algorithms have been thoroughly explained in the course. After running the pipeline, we receive several outputs, including phyloseq-bar plot, abundance table, phyloseq abundance plot, abundance heat map, whose interpretation has been explained as well in the course. The course also introduces the learners to performing downstream analysis of 16S amplicon data using R programming and how to interpret the resulting output. Some of the other example projects that one could look into are effect of dietary fiber intake on microbiome. This example project aims to determine the microbial composition of obese and normal weight subjects and identify microbial changes after weight loss diet. Another interesting project that one could look into is the effect of non-antibiotics and Rett syndrome. This example project aims to understand the changes in microbial community after the administration of non-antibiotic drugs to understand how they affect the neurological dysfunction. So now you might be wondering what comes after the courses, after completing the courses and example projects. And the answer is possibilities are endless. For instance, you could go on to perform a research project like Gus Bhargava, a bioinformatics graduate from Amity University, Rajasthan, who completed a research project on the composition of gut microbiota of samples infected with liver cancer. She uploaded a project on Blown Portal, where an international community of students, peers, and bioinformatics experts can watch her presentation recording, read through the research project, like, share, and comment their feedback under the comment section. Another interesting student project outcome can be seen in case of Dilara Daikin, a molecular biology and genetics graduate from University of Turkey who completed a project on metagenomics and astronaut health, which she went on to present at the Siomics Club session. a session where an international learning community of bioinformatic enthusiasts come together to share their work, debate on important topics, and share their queries to stay up to date with latest advancements in the field. In addition, she also went on to publish her project as a preprint on bioRxiv that she can share with her peers, mentors, and potential employers for future connections. Further, you could also participate in research symposium and showcase your work, such as Omics Research Symposium, where one of the participants, Pawnraj, who presented her project on comparison of microbiome data of different lung diseases, won cash prizes for the first prize for Best Technical Research Project. This symposium is an international research symposium that brings together experts such as Dr. Gareth Rubble, staff scientist at Lawrence Livermore National Laboratory, and Dr. Joseph Borg, associate professor and principal investigator at University of Malta, amongst so many other experts who join as guest speakers and judges. So to view courses and example projects, various programs, student projects, let us begin by signing up on the Omics Logic Learn portal. For that, I'm sharing the link in the chat box. Please click on the link and I'll walk you through the process behind signing up on the portal. So here is the link. So, all right. So once you click on the link, you'll be able to view the Omics Logic Learn sign up page. For users who do not have an existing account on the portal, you can click on the create an account option that you see here. And here you will be asked to enter your name, email ID, provide a new password. You can also have the option to sign up using any of your social media accounts, such as Google, Facebook, Apple account, GitHub account, or even Twitter. So with that, I'll pause for a couple of minutes so that the participants can visit the page and start signing up on the portal. If you are facing any technical issues, please put them in the chat as well so that I can help you for signing up on the portal. Once you have signed up on the portal, please put a yes in the chat box so that I know we are good to proceed. So I'm monitoring the chat box meantime. All right, Nilaba has already signed up. Mansi has also signed up. So we have more than 25 plus participants, and I'm expecting a yes from almost all of them. If you are facing any queries, you could put them in the chat as well. So since I already have an account on the portal, I'm going to click on the login now option. So here I could just click on the sign in and I could enter the course. So once you sign up, you first enter the courses tab page. Here, if you scroll down, and here if it will ask for if you're looking for a specific course. So since most of the participants are interested in the metagenomics field, I'm going to enter the keyword that is metagenomics. There you go. You have the course and the various example projects that you could visit and check, and you could start taking. So one of the important part that I explained before was to set up your profile. For that, click on the tab that says welcome back with your email ID. And here you will be able to see the icon that says profile. Here you will be able to enter your details such as your education background, research interest, your name. And in my case, I've linked my LinkedIn account and my org ID. So under the activity tab, the mentor and the community manager will be able to track your learning progress. Under the courses tab, the participants will be able to view the various courses that they are currently taking. So for instance, I just recently started taking up the example project on non-antibiotics and Rett syndrome. It's a really interesting project, and I would highly urge all of you to check it out. And once you complete a research project like Guzzle, you can upload it under the projects tab. And if you wish to enroll for any of the programs, and then you'll be able to view the programs under the programs tab. And finally, after the completion of the courses, you will be able to receive the certificates under the certificates tab. And finally, to view the various student projects, you can click on the projects tab that you see here, and you could see a wide variety of projects on oncology, neuroscience, agriculture, and so on. So now I'd like to pass on the stage to one of my colleagues, Sonalika Ray, who will be explaining how you can leverage the technical resources that are available on the T-Bioinfo platform. So over to you, Sonalika.
Thank you so much, Shrigari, for the overview. Hello, everyone. I am Sonalika Ray. I'm the bioinformatics project manager here at Pine Biotech, and I'll be discussing about metagenomics, its evolution, sequencing types, several ways to process metagenomic data to pick the right method. I'll briefly discuss about the two major analyses in order to make you understand how easy the analysis could be, and finally showcase some interesting visuals that are obtained by running these pipelines that makes biological interpretation easy and efficient. So first of all, to talk about the connection between disease and microorganisms that has been established in the 1800s. These studies provided important information about bacteria function in isolation. However, it became apparent that most microorganisms live in complex communities, many members of which are impossible to culture in isolation. Today, we know that the microorganisms or the microbiota that make up the human microbiome are not just unicellular organisms living alongside each other, but instead form highly regulated, structurally and functionally organized communities attached to surfaces as biofilms with interspecies collaborations as well as antagonisms that contribute to ecologic stability. However, since the 1990s, microbiome studies were significantly advanced through such major initiatives as the metagenomics of human intestinal tract and the subsequent Human Microbiome Project. Huge studies that have been made possible through the advances in metagenomic sequencing. As a result, skin, lung, gut, and even brain microbiota is starting to be characterized. Each new project represents vast opportunities and challenges to determine which microorganisms play key roles in the development of certain environmental conditions needed for healthy function of human organs and which can be harmful to it. Due to the advances in metagenomic sequencing and other omics technologies, enormous database of sequence data from such communities has led to the emergence of computational metagenomics, which can help study the 99% of uncultivable microorganisms in the context of their natural environment. Unlike other omics, metagenomics does not study a single organism, but the whole community at once, a snapshot of a collection of organisms directly in their natural environment. Since the genomes are so diverse, this is typically accomplished by sequencing a specific marker portion of the genome instead of all the genomic material present in a given sample. And why I'm telling you all about this is because you'll have to work with such data in the duration of the hackathon. Though the sequencing principles vary between different sequencing approaches, to mention the main difference, Illumina specializes in short read-raw sequence data, while PacBio focuses on long read-raw sequence data, and the yield per flow cell of Oxford Nanotechnology is much higher than PacBio because each nanopore can sequence multiple molecules. Now let's briefly discuss about the types of metagenomic sequencing techniques. One is whole metagenome sequencing or shotgun metagenome sequencing. That is relatively new and powerful sequencing approach that provides insights into community biodiversity and function. The other one is 16S amplicon sequencing. That is recently developed technique and is widely used to study microbiome directly in its environment. Such metagenomic sequencing focuses on 16S or 18S ribosomal RNA that has to be amplified with PCR amplification. The 16S gene sequences contain hypervariable regions, which are also known as HVRs, that provides species-specific signature sequences useful for bacterial identification. The benefit of amplification is tied to the nature of sequencing read length, which can maximize effective output from affordable sequencing technologies. To account for the diversity of such sequences, they are usually combined into operational taxonomic units or OTUs, which can be used to classify group of closely related individuals. OTUs are formed from groups of closely related genomic sequences, and they represent a cluster or a phylogenetic clade that has a meaningful abundance in a sample. Such OTU sequences are annotated by mapping onto a reference database such as that of Ribosomal Database Project, Green Genes, or the Silva database. And such databases contain taxonomic information for the domains of bacteria, archaea, and the eukarya based primarily on phylogenies for small subunit RNAs, 16S for prokaryotes and 18S for eukarya. Let's now talk a little bit about the practical analysis approach to analyze metagenomic data on the T-Bioinfo platform. One such analysis is processing the data-to pipeline that is based on running a number of programs, including data-to, ape, and phyloseq algorithms. Data-to generates amplicon sequence variants, or the ASV tables, which are similar to OTU tables but detailed, in that they tabulate the number of identical amplicon sequence variants from different samples. Microbial studies utilizing data-to provide high-resolution, accurately reconstructed amplicon sequences that improve the detection of sample diversity and the biological variants. The data-to pipeline implements a complete pipeline to turn the paired end FASTQ files from the sequencer into merged, denoised, chimera-free, inferred sample sequencing. By using the data-to algorithm, fine-scale variations are identified and a quality-based model is produced. We will end up with merged, denoised, chimera-free, inferred sample sequences. Data-to analysis will filter input data, dereplicate, providing abundances of unique sequence data, and an ASV table, a denoised high-resolution output file. The results obtained from the data-to pipeline includes a microbial abundance taxonomy bar plot, a relative and a proportionate plot indicating the richness and abundance of taxa identified, phyloseq plots based on the OTU abundance, 2D and 3D PCA plots to visualize the cluster separation between the samples of the dataset, alpha diversity measure that summarizes the structure of an ecological community with respect to its richness. Now let's talk about the KIND2 pipeline. The KIND2 pipeline is an open source bioinformatics pipeline for performing microbiome analysis from raw DNA sequencing data. The KIME is designed to take users from raw sequencing data generated on the Illumina or other platforms through publication, quality graphics, and statistics. This includes demultiplexing and quality filtering, OTU picking, taxonomic assignment, and phylogenetic reconstruction and diversity analysis, along with visualizations. KIME has been applied to studies based on billions of sequences from tens of thousands of samples. The advances in the analysis of amplicon sequence datasets have introduced a methodological shift in how research teams investigate microbial biodiversity, away from sequence identity-based clustering, producing operational taxonomic units or the OTUs, to denoising methods producing amplicon sequence variants. KIND2 plugins in the pipeline on the T-Bioinfo server exist for latest generation tools for sequence quality control from different sequencing platforms, data-to and dblur, taxonomy assignment, and phylogenetic insertion, which quantitatively improves the result. The platform provides many interactive visualization tools, facilitating exploratory analysis and result reporting. along with the data to pipeline results, which included taxonomic bar plots, PCA, and alpha diversity measure. Kind2 pipeline also provides lists of results that you could see here on the screen, which includes the alpha diversity box plot, which determines if an environment has been sequenced to a sufficient depth through evenness group significance and faith group significance plots. It includes kind2 alpha rarefaction, which illustrates the number of samples in each group. Kind2 remove eukaryota generates an interactive bar plot of taxa present in the samples. It also includes group significance plots, which include alpha and beta group significance, and kind2 core matrix, an array of alpha and beta diversity measures that can be generated with a single command of kind2. The kind diversity core matrix phylogenetic command produces both phylogenetic and non-phylogenetic measures, as well as alpha and beta diversity measures. And then we have the core matrix phylogenetic command that generates a plot, as you can see here on the extreme right side, that includes the Jacquard distance, that includes the qualitative measure of community dissimilarity, Bray-Curtis distance, which includes, again, a quantitative measure of community dissimilarity, unweighted unifrac distance, and weighted unifrac distance. I hope you will now agree with me that analysis isn't always complicated. There is a logic behind it. And once you understand the logic behind it, then it is easy for you to understand. Today, what I presented was a very brief overview of the flow from the metagenomic study to what are the various sequencing types, and then what are the different analysis types, and showcase you the resources that you could use during your duration of hackathon and utilize. So now to discuss how the resources could be used effectively to build a solution or a product of your own, I would like to pass it on to our CEO, Mr. Ilya Brodsky. Over to you, Ilya.
Thank you, Sonalika, and great to see you all here. I would like to speak briefly about the context, the business context of some of these analysis tools and project examples that that already we heard about. So one of the challenges in bioinformatics in general is that bioinformatics seems to be detached from practical challenges and problems. So the hackathon is an opportunity to connect biotechnology, or where the data is getting generated and what to do with that data, to bioinformatics, where we focus on analysis approaches and insights from data, with real world problems. And these are defined in business terms. So as someone who has started several different companies in the past, has a lot of experience and success and failure in a lot of these companies, I know that there are several important things to think about as you approach this hackathon. So the first one is, what problem are we solving and how are we solving this problem? The second one is, what does the solution really look like and who is going to use that solution? And then what is the business value proposition? How does this solve a business problem that someone is willing to pay for? So the objective of the hackathon is to connect these together and allow student teams or research teams to try and propose some of these problems and face the kind of real world challenges and opportunities that the microbiome has produced. So one important aspect of this is really how to narrow down the many different opportunities that are out there that align with your interests and have the potential to be interesting to investors, users, or companies that you might work with or for. So as someone who maybe comes from a biotechnology, bioinformatics, or a research background, you might be thinking, how can I leverage some of my understanding of this field and what the potential of the microbiome holds to really showcase my skills and understanding of how this could work in a very, very practical way? And so this is going to be critical to understand beyond just kind of the technical things that we've already heard about, and focused on what does the solution really look like. So the first subject that you will have to decide is what problem will you solve? And we separated this hackathon into 3 specific problems. The first one is gut microbiome. And again, we are focused on preclinical challenges and problems. So the reason for that is because when you start going into clinical problems, there's all kinds of ethical as well as regulatory issues that we just don't have the time to address. So this hackathon is going to be focused on preclinical opportunities. So what are some opportunities with microbiome for health and wellness? The immediate thing that comes to mind, of course, is obesity, general kind of health and wellness, but it becomes a lot more detailed as you start looking at the data. So what we will do for that particular challenge is bring you experts that have seen multiple companies that are trying to innovate in that space and connect insights from microbiome studies to specific challenges like obesity or preclinical conditions like pre-diabetic patients and people who experience negative results of obesity, to try and solve them with different types of interventions. And we'll talk about what opportunities are there, what other companies are trying to do, how you might differentiate, and what is the real value proposition for some company or idea or a product that wants to disrupt the market and do something different. The second field is related to beauty, aging, and skin microbiome. So the skin microbiome is very different from the gut microbiome, and that leads to specific methods of sequencing, data preparation, analysis, etc. And so what we will do is we will curate several examples that during the month of November, we will actually share some insights from. And we will bring some experts who are actively pursuing this opportunity with skin microbiome and have identified some interesting trends and maybe special opportunities that you could focus on as well. The third one is related to the general topic of femtech. So femtech refers to technology that can help address issues like pregnancy, healthy birth, conception, and things like that. And so what is important there is that, you need to understand how real data can inform us about these things in general as a research problem, but then also how can we take insights from these research questions and turn them into practical actual tools or products that people will use. So one example of a product could be a recommendation engine. And so we will spend some time speaking about what is a recommendation engine, how can we use insights from microbiome data analysis and link it to other types of information like diet, physical activity, genetic background, and environment. So what's important is to be able to use some kind of analytical approaches to be able to categorize different types of interventions, suggest combination of steps that need to be taken, and also to monitor progression. We will also talk about who is your audience and the importance of understanding your audience and how they view a potential product. So here what's going to be important is to think of, the age range, the background, maybe in terms of where people work, what do they know, what is their education level, and to be able to analyze that data and come up with real clear understanding of what is the market that you're trying to approach with your solution. So we'll talk about some of the challenges and opportunities that existing companies on the market have faced. For example, a lot of the companies that are already making microbiome testing available on the market are facing a problem of knowledge and understanding of what the microbiome can do for them, cost of sequencing and cost of these tests, so how much people are willing to pay for some of these tests, or even their ability to follow the instructions that maybe these tests will recommend, right? Purchase additional pro and prebiotics, change their dietary habits, etc. So then we'll talk about what is the product, right? What really could you propose as your ultimate solution? And that can fall into several different categories. So it can fall into the category of a real physical product, for example, a pre or a probiotic. That could be a pill, that could be a cream, it could be some kind of a marketable product that is physical. And then you face additional challenges, right? Where are you going to source those specific interventions? How are they going to be packaged? Who are they going to be sold through, etc.? Maybe you're thinking of a product idea that is an application that the users will use on their smart watch or their iPhone or some kind of a mobile device. That could be an option as well. And there you need to think about, well, what exactly is it going to do? Where's the data going to come from? And what kinds of insights are going to be made available? And then you could also might be thinking about a product that is not for a customer. It's maybe for a company. So a company will need a very different type of insight compared to a user, right? So instead of an individual and their recommendations, they might want to look at comparison between multiple individuals or multiple types of products or multiple types of challenges. So really, it's a platform kind of an idea. In addition, we will talk about how to define your competition in terms of the value chain. For example, there are many different microbiome testing kits on the market. How are they different from each other? Why do each one of them claim that they are the most advanced or the most comprehensive? How do we actually know if they're more advanced or more comprehensive? And then based on their pricing, how can we identify who is going after the low pricing strategy, who's going after the high and premium kind of a product strategy, and how can you position yourself and differentiate sufficiently from this competition to offer something new, different, and valuable to your target market. We'll also talk about some of the trends in the market, which areas have been tried, which areas remain to be tried, and what is successful, what is not successful, what is growing, what is kind of diminishing. What are some of these trends that are important to look at? Finally, we will talk about how your solution can capture the market. So as a whole, we know that the market is growing at a fairly rapid pace. At the same time, we know that investors are becoming concerned with new companies just proposing kind of marketing campaigns of the same solutions focused on just pricing differences. So really, there's going to be a challenge not only to develop an idea about the product, but also define how can this product be introduced. Now, of course, this is not going to be something that you have to figure out completely over the course of the full hackathon. The idea here is really to think through all of these different stages, understanding the microbiome space from a technology and from the value proposition sides, combining this understanding with a practical opportunity in one of the three areas that we already defined, and then working with a mentor to get enough feedback about your proposal so that you can actually present something meaningful and valuable at the hackathon end. So with that, the key kind of result from all of this is going to be your demo. And the demo is primarily a presentation. So the presentation will address your insights. So this must be based on real data. Again, we will provide you with some examples of data, but we will also talk about other repositories of data that you could leverage to gain insights. And so you can use either the tools that we have described today or your own analysis pipeline. But the main idea would be that whatever your proposed solution is, it is based on insights that are rooted in real data sets. These data sets might not be comprehensive, they might not answer all of the questions, but there's enough evidence that this is realistic and important. The second will be your market research analysis. So how have you analyzed the market itself, the competition that is out there, how many people or companies will be using your product, and what are some trends that are important to understand in terms of regulatory frameworks, opportunities for monetization and things like that. The next point will be about product design. What is this? Is this a physical product? Is this an application? Is this a platform? Is this some kind of a report that you are selling or that you are proposing to develop? And how will that be used by real users? Where will they find out about it? How will they produce data that will be ingested? What would be the results of that analysis? What would be the actual points at which they need to interact, maybe with support or consultation or something like that? So thinking through all of those aspects of the product, so to say. The value proposition. So how is this product solving that problem that people are willing to pay for, right? And what is the alternative that maybe they have to avoid to choose this proposal that you have? Then we'll talk about go-to-market strategy. Are you going to advertise and sell on Amazon? Are you going to develop an app and put it on the iPhone or Android platforms? Are you going to organize some kind of a marketing campaign with hospitals or wellness centers? What exactly is your strategy, and how realistic is that strategy? What needs to happen for that to be successful? And then finally, what is the business value? So what is the business value that is being produced by using this solution? Are these new insights that are somehow valuable to companies? Are these recommendations that people can use? Are they going to stay with it just for one time, or is this going to be a continuous value that is generated and maybe to multiple users at the same time? So really the culmination of this hackathon is going to be about how you can contextualize your understanding of biotechnology, your bioinformatics capabilities, and your kind of understanding of data analysis approaches like algorithms and user interfaces to produce some kind of a business value. And we hope that as a result, you will showcase your ability to think differently, kind of understand the market and propose real innovative solutions, and connect with experts in the field, companies, as well as investors that are ready to consider in a more serious way some of the ideas that you might have. So with that, I would like to pass it on to Ayushi, I guess, to talk about the schedule of the hackathon and the registration, or who is going to cover that?
It would be me, sir. So let me share my screen for discussing that. Okay. Is my screen visible? I'm unable to share my screen. One moment. Okay. So is my screen visible now? Okay.
Yeah.
Okay, so in this part of the presentation, I will be discussing about the program registration details. So as discussed, I'll just quickly recap the resources that you get as a part of this a hackathon. So first of all, you will have access to the research and computational infrastructure through the T-Bioinfo platform. You will also have access to the Omics Logic training resources that comprise of online coursework, support, and mentor guidance. And finally, it will also comprise of a practical example project, data sets, case studies that we have discussed in our previous slides. And finally, you will have access to expert workshops and mentor guidance The week 1 workshop will be on microbiome business, and the topics covered will be microbiome health and wellness, omics and microbiome technologies, bioinformatics for health and wellness. And the second week of the workshop will focus on biotechnology topics such as gut microbiome, skin microbiome, and vaginal microbiome. And the week 3 workshop will be focusing on the computer science topics. That will be for infrastructure for computational analysis, tools, methods, and algorithms, machine learning, data visualization, and user experience. So the main event will be conducted from November 23rd to November 28th. In between this period, you will be required to develop a minimal viable product, and you will be able to present during the main event. And with that, you will be also able to receive certification and recognition and exciting crash prizes and funding opportunities. And for the speakers and the judges who will be participating, we have a wide network of experts who will be participating, such as Ms. Liliana Nordbach. And here are some of the important dates that you need to keep in mind. That is, the registrations has been open on September 30th, and it is still ongoing. The registrations will close on October 31st, and the hackathon training program will start from November 1st, and the hackathon event will be held from November 23rd to November 28th. And to get all the details that we have discussed in this webinar as well as the previous webinar, you can simply visit the main hackathon page that you see here. I am pasting the link in the chat box. So here, if you just click on the button that says register for the microbiome and health and wellness hackathon, you'll be redirected to a form. Here you will be asked to enter some of your details, such as your name, email ID. And once you fill up the form, you'll be able to get all the latest updates that are happening with the hackathon. Now, coming to the registration cost part. So once you scroll through here, you will be able to view the registration cost for different categories of the participants. So if you're an individual participant, you can register for $100. And if you're participating as a group of students, you can register for $300. And if you're a student from one of the partner universities, you can register for $100. So to know if your university is a part of our partnership program, you can simply visit the link that you see here. That is the main page for the Partnership University program. So you can browse through and check if your university has been listed under our Partnership University program. And then coming to the payment checkout. So each of it has been divided in different categories. So here, if you were to click on the respective category that you're participating, you'll be asked to enter some of the main details. And once you enter all the main details, you will be able to make the payment, and you'll be able to register for the hackathon. So with that, if anyone have any queries so far, please put them in the chat box, and our team will be happy to help you. I'm also sharing the contact details of our marketing manager, so that if you're having any queries, you can reach out to her at your own pace. So there you go. So we'll just wait for a couple of minutes so that if participants are having any queries, we can take them up. You can browse through the main page at your own. Okay, Nilaba, thank you so much for this. Excited to participate. Yes, we are excited to have you as well. So all the very best to Nilaba for participating in the hackathon. So in the meantime, while we are waiting, let me just briefly navigate through the main page, hackathon page. Here you will be able to see the various topics under which the hackathon are based on, such as obesity and precision nutrition, skin and beauty products, pregnancy, and femtech. And here, once you scroll down, you're also able to see the various topics that will be covered during each of the expert mentor workshops of the 3 weeks. And you can also browse through and see the various speakers who will be participating, such as Ms. Liliana Norbach. She's a serial entrepreneur and investor in high-tech biotech companies with a demonstrated history of co-founding and growing several companies with successful exits. So if the participants are participating in the hackathon, it's a really great opportunity I would highly urge all of them to participate in the hackathon. So we're waiting for the queries. If anyone have any queries, you can put them in the chat. Or you can unmute yourself and speak as well if you're having any queries. Otherwise, we are good to end with today's meeting. And I hope to see you all for the next webinar that will be conducted on October 27th. So with that, I'd like to end today's meeting. So thank you, everyone. Have a great day. Okay, Stephen has raised his hand. Yes, Stephen.
Yeah, thank you so much for the presentation.